So welcome everyone. First of all, so we're really happy to be seeing you here today. So my name is Ibrahim Serdar Koçar. I am a project manager working in İzmir Commodity Exchange, and we are gathered here today for our project, like innovative sustainable cluster for olive value chain, like shortly OASIS project, that we are implementing with participation of Siambari, as they are the hosts today. So I would like to thank like Siambari and first of all like Ana Maria Dongia, Dr. Ana Maria Dongia and the Siambari for their initiative for this webinar happening today. That with the initiative now we are organizing this webinar. And I think we can start slowly. Our new members are also coming, new participants are also coming. I will be like keep accepting them. But since here we can start, and I would like to give the microphone and the speech to Dr. Ana Maria Dongia for introducing herself and also starting the presentation slowly. Thank you so much. And first, before that, sorry for interrupting. For the reporting purposes, the meeting is gonna be recorded for your information. So if you would like to have your camera open, that's your opinion, but you can keep your cameras closed. It's already closed, like as the setup. But also for your questions, like you can write it from your chat and from the questions and answers part. I will be checking it continuously. And we can also direct to Dr. Ana Maria Dongia when it's necessary. Thank you so much. Sorry for interruption. The microphone is yours.
No, thank you, Serdar, and thank you for this opportunity, because I mean Siambari is a partner in OASIS, and of course we are also suffering the threat of Xylella Fastidiosa on olive trees in our region, and for us it's really a responsibility to highlight this serious threat for olive growing areas, not only in our region, but I should say in the rest of the Mediterranean. So I'm just saying that Xylella Fastidiosa, of course I'm working as a scientific area coordinator, but I am a plant pathologist. So of course my first, let's say, interest is on pathogens. So Xylella Fastidiosa olive quick decline syndrome, and not only, but primarily, of course, because this is the major threat to olive. But how to prevent or live with this serious threat? First of all, we need to know what is the threat. And so protecting olive growth begins with understanding the threats that challenge their health, resilience, and productivity. And this is what will be about my presentation. Of course, I acknowledge CNR Bari, which is a scientific institution that is also, as Siambari, involved directly in this on this program for Xylella Fastidiosa, and Apulia region, which is has also a specific website dedicated. Before to start, I would like to let's see if it goes. Yeah, to just spend really few words about Siambari, because for people that do not know, Siambari is one of the 4 institutes of this organization, which is SIAM, Centre International de Haute Études Agronomiques Méditerranéennes. And in general, we promote sustainable development, agriculture, fisheries, food systems, in order to improve food security, optimizing One Health approach. And the 3 major tools are education and training, applied scientific research and innovation, development, and scientific cooperation. Of course, about plant health, we have a very long experience, I should say more than 35 years, in order to face problems of pests and diseases affecting food security, especially in the Mediterranean region, and through the promotion of scientific innovation, so advanced training, international cooperation. Our major, let's say, contribution is on developing and transfer advanced diagnostic methods, tools, characterization, epidemiology, surveillance, and modeling on priority pests, but with a special emphasis on quarantine pests, and affecting, of course, major crops of Mediterranean importance. And this is, so our goal is to raise awareness, and this is one of the, today is also one of our goal, and develop knowledge on what are innovative methods and tools for pest detection and control, integrating smart technology. This is also very important for implementing precision decision support systems. And of course, Xylella fastidiosa is one of the priority pests we work with. But we have also another function. We also provide scientific and technical support to the Apulia Plant Protection Service and also to other National Plant Protection Service of organizations in the Mediterranean region, and through proactive measures. I mean, how to apply monitoring programs for quarantine pests and certification of a plant propagating material, which is a healthy material, so pathogen-free. And of course, Xylella fastidiosa is always a priority pest in this. So we have, for this purpose, we are accredited quarantine center for manipulation, maintenance of plant quarantine pathogens, accredited diagnostic lab for official testing of regulated pathogen. And also we have these functions to have a repository of primary sources. I mean, we also, through a pomological sanitary selection program, try to produce pathogen-free mother trees of mainly major fruit tree crops. And now, going to Xylella. Xylella fastidiosa, in reality, is a very old problem. You see here in the 1880s, so we are really going back in the last century, because it was found as a severe disorder in California on grapevine, and after, during the years, it was found. I mean, the name of the disease was grapevine purse disease, but until it was really recognized as a bacterium, it happened really in the 70s, 80s, so quite recent compared to the first finding. And in California, so after 125 years, the purse disease, which was the major disease caused by this agent, is still the most severe disease of viners in USA, and not only in California, but also Texas and Florida. So this is just to give you the idea that it's not a simple problem, because if in this states of USA, where they have a lot of funding for research, a lot of support, they have not found a solution. So you can imagine how critical is this issue. So this is very important, because people think that solutions can be found rapidly, but this is unfortunately not true. So what is the agent? As I said, is a bacterium, is not sporing bacteria, but is a fastidiosa because it's very slow growing in arsenic culture. So the culturing is very difficult, and is localized in the xylem vessel, so very inside the vessels, and that is why it's also very tricky and difficult to control. I just want to give you the idea that in reality there are different subspecies of Xylella. So we have the subspecies. The major one are Fastidiosa fastidiosa, which is the major agent of Pierce disease on grapevine, and then we have the Pauca, which is the major agent of the Citrus variegated chlorosis in Brazil, and now recently on olive trees in our region. And then the multiplex, which is very widespread. And we have the minor ones are the Morus essendi. Now, what is really amazing, that even if it's one pathogen, the host species are different depending from the subspecies of this bacterium. So just to give you an idea, this is not even updated, but the Fastidiosa fastidiosa, which is affecting grapevine, for example, is not affecting olive trees as a multiplex, and the multiplex is not affecting grapevine, and the Pauca is affecting citrus, olive, but not the grapevine, and so it's it's acting, in reality, each subspecies is acting as a different pathogen, even if the pathogen is the same. So its natural host range includes 71 families, 210 genera, for about a total of 463 plant species by now. And here, just looking to this map, you see the majority of the findings is in the American continent, north and south, but we have now, unfortunately, Europe, especially Italy, Spain, and France. And then we have two here, we have Israel and Lebanon, and then Iraq, Iran, and China. So it's a very difficult, again, task. And we have the causal, as I say, the major problem, the first major problem of grape was Pierce disease, of grapevine, but in reality, the causal agent is affecting many crop, ornamental, forestry, and wild species. And based on the species, we have also different diseases, like we have here the Pierce disease of grape, the citrus variegated chlorosis, which was also found many years ago in Brazil, and then the almond leaf scourge, plum leaf. So depending from the species, we have a different disease. So the number of diseases is very long. And what is important to say that the long distance spread of this pathogen is primarily through human-mediated movement of infected plants. So trade or transport illegally plant material for even for ornamentals, not only, for example, in Europe, we believe, because we have traced back the introductions that was introduced through ornamentals. Millions of ornamental plants were introduced from Central America, where the problem was in ornamentals. So at the landscape level, I mean, once arriving in the area, the spread is mediated by xylem-feeding insects belonging to the order Hemiptera. In reality, here, I just mentioned the two major ones, which is Homoloadisca coagulata, is the sharpshooter, which is vectoring grapevine Pierce disease, primarily in California, in USA. And we have in Europe primarily Phylloemus spumarius, which is the major vector. And but in reality, every time it invades new areas, all xylem sub-sucking insects could be potential vectors. So this gives you the idea that has not really a specific vector. So it's and the vector plays an important role because the bacterium doesn't move without the vector. So the spread is mediated by the vector. And otherwise, it stays in a plant without moving. So Phylloemus spumarius, which is the most efficient vector of the subspecies Pauca ST53, which is that one that is affecting olive trees in our region, unfortunately is polyphagous, so it's not only transmitting to olive, but also to other species, and widely distributed in Europe, and has a very high population in warm areas, as it is in south of Apulia. So it's a very tricky vector. And well, the first outbreak occurred in 2013, so now are already 13 years since the first finding, and was, of course, identified as a disease named olive quick decline syndrome. And on that time, it was in the APOA1 list of quarantine organism, which means there was not spread and was not present in the European territory, but now is in the A2 list, which means that is present, and in some areas is also largely distributed. So it's always a quarantine organism, but is in a different list. And I want just to highlight that it occurs here in southern part of Apulia, where the red circle is here, this is Apulia. And as you can see, the green, it's the mapping of all olive growing areas. So as you can see, the whole Apulia is, let's say, grown with olives, and the south more than in the northern part. And so what occurred as first finding was, I should say, the major olive growing area as ancient olive trees. And unfortunately, the ancient olive trees were the most affected and susceptible. And soon after the first finding, the Commission implemented decisions regulated measures to prevent introduction spread in the Union of Xylella Fastidiosa. So in order to regulate early detection of outbreaks, harmonize survey activities. And these regulations are very stringent because the problem is very serious. And unfortunately, it's serious because there are no effective control measures. So this is a very critical issue. And so we have in Apulia region 40 host plant species, which has been identified as a host of Pauca ST53. And recently, we have found also the Xylella Fastidiosa in the northern part of Apulia, which is ST1, which is the agent of Pierce disease or grapevine. And then we have found the multiplex, which is affecting almond, cherry, plum, peach, and the Spanish broom. So in Apulia, we have the three major subspecies. As a long period of latency, so means that we don't see symptoms soon after the infection occur, and some species, some host plants may remain symptomless for years, and this was some of the problems in ornamental plants, and so it was very difficult to see the problem in plants that are not for crops. So for crop use, so in olive trees was very evident because centennial plants were dying. So of course you have to look what is. But if is dying a polygala that is a common ornamental, nobody is caring, no? So this is what happened in Corsica, for example. In Corsica, the polygala was dying, but nobody was caring about because was not very important. But then when they found out the polygala was a host of the strain of Xylella occurring in Apulia region, they tested the polygala, they found infection in course. So it's very difficult to identify infections soon and can adapt to different climatic conditions. This is a very also critical issue. And as a long list of vectors, because as I said, in reality, all sub-sucking insects could be vectors, and most of the vectors are polyphagous. So the methods to control are only based on eradication of infected trees and vector control. Vector control is very, very important because, as I said before, the vector is the only way for the bacteria to move. So the role of the vector is very, very critical. And so this eradication vector control, of course, is mandatory in the demarcated area, which is including buffer and containment zones. Here we have just some ideas of what happened. I mean, eradication, of course, and also the burning of the material that has been eliminated through the eradication on site in order to avoid the risk of moving the insects in other locations. It was nice to find out, fortunately, that the species are not susceptible to these subspecies Pauca ST53 are grapevine, citrus, peach, apricot, and plum. And this is very important because now they can also be planted in the affected area. except those ones where eradication measures are in place. As you can see here, this is the olive trees with the Xylella symptoms. Here is the grapevine, which is not showing symptoms. The same here for citrus groves close to the and infected, in Xylella dying for olive trees dying for Xylella symptoms. Of course, if someone want to ask questions during also I'm open, okay? I want to show now the European situation concerning the subspecies, which after 11 years of mandatory monitoring programs in the EU territory, we have seen that we have almost all over Xylella from Spain, France, and Italy. And we have a different subspecies, so multiplex fastidiosa and pauca. The pauca affecting olive trees, the ST53, is only located in south of Apulia. It's different, for example, from the pauca here in the Balearic Islands. The fastidiosa we found is that one affecting grapevine, unfortunately, so the agent of Pear's disease. And then we have a different multiplex sequence typing from Italy and all Spain and France. So this is the situation, and as I said before, going to the Mediterranean region, we have in Lebanon and Israel also the fastidiosa, which is the agent of Pear's disease of grapevine. Now going to Apulia region, to Puglia, if I say Puglia, sorry, but Puglia in Italian, Apulia in English. Sometimes I move from Italian to English, but it's the same region. So after the first outbreak, which occurred in 2013 in a very limited area, which is here, which is just at the end, let's say, of the region, because the region is very long. So was just at the end, let's say, of the hill of the Boot, which is Italy. Then now moved up to the north. Of course, in a different way. And if you see here, we have all the red part, which is the southern part of Apulia, where we have only the pauca ST53. We don't have other subspecies. Then we have, you see this yellow band here is the containment zone in the infected area, and the blue area here is marked the blue is the buffer zone. After this, we have the other foci, and we have the multiplex in this foci here, which is Bari province, Taranto province. And then we have the pauca ST53, which is also moving to the north. So this is the updated situation. And what is important is also to highlight two different scenarios. As I said in the beginning, we have the infection in the southern part of Apulia, and all the rest was negative. Now we have, after 1,400,000 plants analyzed in 12 years, and also eradication of many trees, we still have two scenarios. So the southern part, only one subspecies, the northern part, more than one subspecies, but is the spread is much less. And based on these two scenarios, we can say that also there is a mitigation of epidemic in the south, where the olive trees is the primary host, of course, because it's the pauca strain subspecies. And then we have in the northern part a slowdown of the diffusion. So two different, let's say, situation, even if the pathogen is one pathogen, of course. So even the spread over short distances, so the new outbreaks in the north is much slower than it was in the south when it started. It was very rapid. Now, there are reasons why, of course. We can say that the slowdown of the northern spread is, first of all, if you see to this map, the red marked area is the more suitable area for Xylella and also for vector population. I don't know if you see my cursor, yeah. And so going to the north, it's less, let's say, suitable, which doesn't mean that is not suitable at all, but is less. Different land management. Now, this is a very important aspect because in reality, there is also different growing of olive trees in the south and in the north of Apulia. In the south were more centennial trees, sometimes abandoned, so not well managed, without irrigation. So trees were also suffering from drought and from other problems, and the fact that they were not, as you can see here, also soil, land management, which is very basic, and the normal treatments for the olive fly, as it is happening in the north of Apulia, was making different the situation also of a vector population, which was very high. So plants were centennial plants, most of the plants, not well managed, without irrigation system. And so there was also no tillage, so very good environment for vector population. So the situation was very difficult in the south compared to the north. And application of containment measures that has been adopted since the first finding in the south also. So monitoring and removal of infected plants, the vector control, which were intensified, of course, slow down the spread. And so in principle, there is a less abundant vector population in the north compared to the south. And this is make a difference also of spread. And if you see here, as a juvenile stages of the Philenus spumarius, if you see the average of this population density in the north compared to the south, you can see how different is the population in density. It is very different. And also there is a reason why a drastic reduction of reservoir inoculum in the southern part. Being the southern part, affected by the disease. So there is a massive reduction of vegetation due to the disease itself, because the plants have no any more biomass. Significant removal of severely damaged trees, and so replacing with resistant cultivars, and also high frequency of fires in olive groves because of the dry plants after the disease, but also because not accidental fires, but induced fires because they wanted to remove plants without spending money. And prohibition of planting highly susceptible cultivars. So means that the replacing with the cultivars which are tolerant or resistant instead of susceptible, of course, changed also the inoculum quantity of the pathogen. And here we can see that on the contrary of what we have seen before, that the population of the vector is less in the north compared to the population of the vector in the south, going to test the pathogen in the insect, we see that the inoculum is much less in the south compared to the north because we are removing, replacing, and plants inoculum is disappearing. And so even the vector have no more anymore, let's say, the possibility to acquire the pathogen. So is on the contrary, as infected insects, vectors, is less in the south compared to the north. Of course, there is a different status in south and north of Apulia, and we can see that in the southern part, so which is this yellow part, new symptoms of desiccation are milder and less frequent. This is a very important issue. So there is endemic, is epidemic, endemic, but the type of symptoms are not so severe like it was in the past, and more evident in Gelyna compared to Oliarola, which are the major two susceptible cultivars for olive trees. In the northern part, which is the red area, the disease remains active and continues to progress. So this is also the inversion, let's say, or tendency. So less frequent as foci of the pathogen, but the high disease severity compared to the south, where it's almost spread all over, but the disease is less severe. So the mitigation of epidemics, so in the south of Puglia, where we have the problem mainly on olive trees, is that the inoculum reservoir has been substantially reduced. The vector transmission efficiency has declined because there is less inoculum, and the incidence of superinfections has decreased. So we have the disease, but not so superinfection as it was in the first years. And but in reality, there is no change as by the genetic point of view, because bacteria population remains represented by ST53, so the subspecies Pauca, ST53, and also the bacteria titer in plants have not shown any significant reduction. So something is occurring either in the defense system of the plant, which are more efficient in disease expression, and then or there is a sort of a cross protection. I mean, this mechanism are not known yet. As economic impact, of course, there are some articles that are focusing on the fact that there are millions of trees, of course, dead or compromised. I mean, they are not anymore productive, and there is a loss of 132 million euros per year in profitability, and also in work per year, almost 1 million of hours of work per year. And so there is a big impact by the economic point of view, of course, but not only by the economic point of view, because we have had probably now it's much less, but in the beginning we had a really I should say impressive negative perception among civil society, and I want to highlight this because this was one of the major problems to face. And this reduced a lot let's say, the time for action. When we have problems like Xylella or other quarantine pathogen, the tempestivity is important in the actions, even strong actions eradicating plants. But if you want to succeed, you have to be very rapid. Unfortunately, this was in the beginning, the perception was so negative that civil society, most of the civil society, of course not all, were against science. So they were not accepting this problem. And so this was a serious, also as a serious impact also in the application of phytosanitary measures. Of course, there is a decline in land value, reduced attractiveness of tourist facilities, climate impact, abandonment to rural areas, increase in summer wildfires, loss of religious traditions, which is very important also because most of the olive trees, I mean plant material for Easter, for religions were coming from that area. And of course, now cannot be moved anymore. So, and this was another problem. So here there is some ideas of what means to have Xylella, this type of subspecies of Xylella affecting olive trees. Now, this is in the south of Apulia. Let's go and see what is happening in north of Apulia. I mean, after the demarcation that we have here from the south to the north. I hear someone say something, I don't know.
No, I think like the microphone was like open, like for mistake,
okay.
so I just like turn it off, no problem.
Okay, thank you. So going to the north, of course, we have a different situation because we have a few foci isolated, like you can see here, and of the three subspecies. But the current EU regulation is imposing stringent control measures as they are doing in the south, I mean, in the highly infected area. So there are no differences in the regulation as a control measures, even if the impact of this foci is much less than what is occurred in the southern part. And so let's see what happened after 18 months since the first finding of Xylella fastidiosa multiplex. In reality, the impact, that's why we are, focusing in reality on the impact of each subspecies, because in reality, again, Xylella is one pathogen, but the subspecies makes the difference. So the impact of bacterial infections appear very negligible. Symptoms are not specific, can be even confused with physiological or non-irrigated, for example, in almond groves. The outbreak was discovered only because we were doing surveillance program. Otherwise, nobody was caring about the symptoms because they were associated to physiological problems. And besides almond, the risk of infection in other specified plants, host plants, appears very low. So it's a very unclear, the boundaries of affected area, and we think that is an old introduction that is now showing up, but probably because we found the problem through, again, the monitoring program of the pathogen. Otherwise, nobody was, seeing this problem in these host plants. So we think that if we do a mandatory eradication program, as we have done for the subspecies affecting olive trees, which was very severe in olive trees, so very impactful, we will have a severe impact in terms, of course, of profitability and the rest, because we are dealing with grapevine, almond, and other plants, but mainly grapevine. So after 20 months of monitoring of Xylella fastidiosa, which is that one affecting almond and grapevine, again, it's negligible in almond, but in grape, apparently is now affecting only the cultivar superior. But there are no the other cultivar, they are not showing any particular symptoms. So this is the situation. Of course, the situation is very complex. Of course, it's not easy to manage because we have so many different situations in the south and in the north, and especially in the north of Bari because of the three subspecies. So there is a disease prevalence is influenced by the interaction, which is a very complex interaction between a pathogen genotype, host susceptibility, and vector-mediated transmission. Even the vector probably prefer as a dominant, even if it is able to acquire all the three subspecies, but probably inside the vector for transmission, one subspecies is predominant compared to the other. So all these are under study because are all aspects that need to be investigated. As a general conclusion, we can say that significant changes have taken in the evolution of the infection since 2013. And the complexity of the situation, which is different from each subspecies, suggests the need for targeted case-specific management measures instead of a uniform one-size-fits-all strategy, because in reality, the regulation, it's one regulation for all subspecies, so there are no differences. But in reality, we could see by this first finding and monitoring and results that there is a difference among the subspecies, and so probably it's time to consider the revision of the EU regulation. Do you have some questions until now before to go ahead?
I don't see any questions in the chat or like questions and answers part, but if there is any question, maybe like they would like to, the participants might ask like in person now if they want to.
Okay.
But I don't see any hands either.
Okay, I continue. Now, surveillance. This is a very important, of course, to find this, let's say, outbreaks. We need, of course, to have a good surveillance program, and we have guidelines issued by the European Food Safety that is authority that is, let's say, standardized, harmonized these guidelines, and just to give you the idea, very light, that we have a cartographic grid, and each grid is over 1 hectare, is sampled for the main host species. So if we are dealing with the pauca or fastidiosa or multiplex, of course, if a pauca is the olive tree, is the primary host to be investigated. If it's fastidiosa grapevine, or multiplex, the almond. So we first monitor in the grid what these plant species are showing symptoms. Here we have the leaf scorch, for example, in these olive trees. But we may also collect samples as pooled samples from asymptomatic trees if we don't have symptoms. So we can either collect from symptomatic, either from asymptomatic trees. Just to give an example, that in each of these areas, so the yellow is the containment area, the blue is the buffer zone, and then we have the isolated forchi as we saw before in the northern part, we have different measures for eradication. For example, in this zone, we only eradicate the infected, the trees found infected, while if we go in this other area, we need to eliminate also all plants around the infected plant, even if they are not infected, around 50 meters. So it's there are different, let's say, measures depending in which part of the demarcated area you are. But I want to focus the attention on vectors, because as I said, vectors is the major, let's say, problem for spread. Here we have summarized the life cycle of the vector in order to know when we have the juvenile stage, when the vector goes on the olive trees and goes from one tree to another, and then goes back to the wild vegetation. And why we need to know this? Because first of all, we need to monitor the juvenile stage here. I mean, when we have the juvenile stage, it means that the insect is not able to acquire the infection because it has not visited the olive trees, and also is not able to transmit. But we can, once we see that we have these juvenile stages, we can identify the optimal timing for tillage, which is one of the mechanical control methods against the juvenile stages. And so reduce, let's say, the population of adults. And for this reason, it's done in the period of March and May, with this, before goes the juvenile stage, becomes adults and go to visit the olive trees. Then we have the adult stage, from May to October. And these vectors are persistent, means that they are able to transmit the pathogen during the whole adult cycle. So from here to here, it's able to transmit, to acquire and to transmit. And so to know the adult stage is important because the seasonal dynamics of the population of the vector enable the identification of optimal timing for chemical or biological control. So we have to rely also not only to mechanical reduction to tillage of the young stage, but also to the control directly to the adult stage. And the adult stage is the stage where the vector can acquire and transmit. But it's also important to know this data because we can also know when the insect is able to be infected because has acquired the infection. And we call spine sex monitoring because we can test the insects in order to see if the pathogen is in that area that is apparently asymptomatic, but if we found the pathogen in the insect, it means that the infection is already there. So it's important, this type of monitoring for these two purposes. So for the control of the insect and for the early detection of the pathogen in the apparently asymptomatic area. And what we call the spine insect approach. By this approach, in reality, the new foci north of Apulia, I mean in Bari province, of Xylella fastidiosa and multiplex have been found thanks to these insects, the monitoring of spine insects. Because there were no symptoms, which were alarming, as it was for olive trees in the southern part. And but there were routine monitoring program. So by collecting the insect in different points and testing by molecular detection, directly the insect and testing, we could see if the insect was healthy or infected. If it was we found the infected, means that around this one or this point we found infection, we have to search around on plant material, which is apparently asymptomatic, and in this way new foci were found. So it's a very important approach to indirectly assess the presence of the infection in apparently asymptomatic area, so for early detection before symptoms develop. Usually for pathogen detection, we could use serological testing, but in reality in Apulia, everything is a bimolecular assay, following the EPO protocol, primarily real-time PCR, but we have also the real-time LAMP, which is also used for on-site detection. And the institute, I mean, we have also improved the LAMP detection, either for insect, either for plant material. For insect, by testing directly the insect in the tube, and for plant material, by printing the plant material on a membrane, a nitrocellulose membrane, and using a disc for testing. Both technologies allowed the movement of samples, so plant material and insects in printed membranes and in tested tubes, without moving the plant material, which is also risky, even if you are going to close in a bag and to send to the laboratory for testing, but is always risky to disseminate, to spread the infection through insects, because we may carry accidentally the insect vectors infected in new areas. So in order to safe move the samples, this technology can also be very effective. Yeah, the membrane. I want also to show, as SIAM always, innovation, that we think to rely on a multi-source IoT surveillance system, which is still at the prototype stage, in order to early reliably make a surveillance which is early, first, reliable. Because we have to be very rapid in outbreak detection and timely in the implementation or eradication containment measures. That was missing, unfortunately, in our context because of different, let's say, problems for communication with the civil society. And so this system in reality is the integration of different technologies. So we have a satellite, we have remote sensing, proximal sensing, we have statistical basis sampling schemes, and so IT tools like the applications and modeling for forecasting the right period for the insect vector. So it's based in reality on different integration of ICT, geomatic, diagnostic, and predictive tools, and collects, harmonizes, and streams heterogeneous data to a dedicated platform, which is here, enabling real-time spatial-temporal monitoring of Xylella infections and supporting, which is the most important part, data-driven decision-making for the control and for optimizing also the monitoring strategies. So we have a large-scale monitoring by satellite, small-scale monitoring by drones. The application for ground inspection, so for field-scale monitoring, which include monitoring olive growth, spy insect monitoring, and the monitoring of sentinel plants. I will tell you for what about. And then sampling are tested by molecular detection, and all this data will provide, of course, very good support for measures to be applied in a specific area. Going in details, I just want to focus the attention that through the Copernicus program, which is the Sentinel-2 imagery, for example, it's very interesting how can we evaluate in the, for example, here from 2015 to 2023, the evolution of a certain area of the disease. So we can see here that as a progressive loss of biomass, for example, we can see that in the olive growing areas that the red tones indicate a low or no biomass, which is what is happening once the disease is already spread and severe, compared to the green tones, which represent higher biomass. So here the plants were showing higher biomass because they were not showing the disease as much as in the 2023 after these years, because the disease was progressive, so there was a loss of biomass on the trees. And so we can have indications based on this spatial and temporal evolution of the disease of something occurring on the ground to be inspected. Then we have also the small-scale monitoring, which is based on a specific sensor that was developed for the purpose of investigating for Xylella Fastidiosa, and is based on the use of a specific package. So it's a web-based multispectral sensor prototype that is based on spectral bands, which indicate the presence of Xylella and asymptomatic olive growth before symptoms development. In order to identify these spectral bands, we had to combine two methodologies, which is the spectronomic approach, spectral radiometry plus metabolomic. Now, by combining these two approaches, we could identify metabolites which are produced by the plants when it's infected at the very early stages. And we can also identify the spectral signals of this metabolite And based on these spectral signals, we can set our sensor mounted on a drone in order to make the monitoring of Xylella fastidiosa, especially in pathogen-free area, in a symptomatic area, when we don't have symptoms, but we may identify the infection in a very early stages. Now, this is still at the prototype stage because it's the outcome of a project, but it will be very effective for the plant protection service in order to investigate from a drone the olive groves. At the field scale, we have to rely on application, and this application it's able to enables data collection, storage, real-time transmission of the data collected on the ground, and integrates also what is indicated by satellite or web imagery as suspected areas or groves to be tested by ground inspections, but is also based on statistically sampling schemes to be applied in olive growing area in order to have the higher efficiency of sampling with the less number of samples. And it combines data for the spine insect monitoring, which have been already described. But we add another component, which is very important. It's a sentinel plant network. I mean, these plants are a host, of course, susceptible species, but they are very easy to grow, even on pots, and could be also placed in the risky points, as could be airports, seaports, nursery, in order if something is arriving from these borders, risky point, they can get infected and symptomatic soon, and could reveal the presence in a very short time of the problem, in this case of Xylella disease. And but they also have a second role, which is they also attract the vectors. So means that the inspector can go inspect for symptoms, but they can also collect the vectors and see if they are infected or not by the pathogen, because in that case means that if the vector is infected, we have the problem close by. So we have to investigate. So that's why I call the sentinel plants. Of course, this is the whole system, and we have already seen the lab detection, and this is we hope to I mean, some components are already operative, but other components like this one, the prototype here, and for example, the sentinel plants are not already in place. I mean, operative. The last part is our measures to live with Xylella. I want to highlight that there is an authorization in Apulia to live with Xylella by planting not only olive trees of the resistant tolerant cultivars like Lecciana, Leccino, and Favolosa, but also the specified plants, I mean plants that are hosting also other species as almond, cherry, citrus, peach, plum, grapevine, apricot, and other species like rosemary, myrtle, etc. So as you can see, the Lecino, for example, which is a resistant species, must replace, unfortunately, because there are no other choice, the Oliarola or the Cellina, which are the most susceptible species, varieties, sorry. And but it's not enough to rely on tolerant, resistant, or let's say not infected species, but for planting. But we have also to apply agronomic measures for vector control, because again, the vector is the most important actor. And the pruning is very important, so it reduces infection opportunities, especially removing the tender shoots, which are easy for the vector to penetrate, and then optimize also by pruning the use of plant protection products for distribution. And it's also a good practice to remove the suckers, because they are usually very soft and they can be attracting for the vectors. And also the pruning residues must be or shredded on side or even burned. So in order not to move material, which can be always dangerous. And as agronomic practices, of course, tillage is very important, especially in spring, against the juvenile stages we have seen before. And also to keep the soil free of weeds, because weeds can host the vector and so the pathogen also, and the vector can visit the weeds and transmit to the olive trees. So we have to keep the soil free. And flame weeding is permitted only where you cannot move with the tillage or herbicides, but always considering the environmental impact. So now in the 2026, mechanical control of vector is mandatory in the whole Apulia region. But in general, we can say that is strongly recommended in all pest-free zones wherever you cultivate olive trees. So this is something that I want to highlight, especially for producers, olive growers, that this practice is very, very important. And also the use of insecticide for the area where we have containment measures. We have the need of 2 phytosanitary treatments on olive trees, which is mandatory in the buffer zone and containment zone, but is recommended also in the pest-free zone. And should not be performed on completely dry olive, almond, cherry, and grapevine trees, in green areas, in urban areas, in woods and pine forest. And here we have what has been issued by Apulia Reggio for the control of olive, for the control of the Philaenos spumarius on olive trees. We have 6 active substance and mode of actions, and of course some limitation in their use. And it's very important to do this treatment once is starting the adult to go to visit the olive trees, in order to catch the adults before it acquire and distribute mainly the infection. In general, we can say the contingency plan, so describe for Xylella Fastidiosa, describes measures to be taken to prevent pathogen introduction spread, and as well as emergency response procedures if the pathogen is unfortunately detected or suspected to be there. So when we have to adopt measures against Xylella, because Xylella is present, so we need to delimitate infected buffer zone, intensify surveillance diagnostic testing, eradicate or containing infected plants, control insect vectors, communication protocols, coordination among authorities, communication to civil society, measures to prevent further spread. I just summarize, but it's much more complicated than what I'm saying in these few points. And living with Xylella, we have seen that in the southern part of Puglia, where we have already Xylella spread in all that area, so the use of olive-resistant tolerant cultivars as certified plants already Xylella-free. So we have to be sure that we are planting material that is certified, so pathogen-free, but has to be resistant tolerant cultivars. But not only, we need to monitor and control insect vectors, to target agronomic practices and phytosanitary treatments, and implement also good agronomic practices in general as balanced fertilization and proper irrigation management to enhance plant vigor and resilience. Because of course, if we irrigate olive trees, so we don't induce stress in the plant can also react better to the infection if the inoculum is not so high. So it's very important, the good agronomic practices in general, in particular those ones that are targeted for the control of the vectors. And but even waiting for Xylella, I don't know if it is nice to say waiting for Xylella, but in all other pathogen-free areas, besides the recommendation of the same measures we have seen here, so implementation of good agronomic practice, monitoring control of insect vectors, use of tolerant resistance olive cultivars for the new planting. Of course, we have to add the disease pathogen surveillance at risky points and the good communication of in the surveillance program with the citizen science. And so we recommend, but in reality, I think olive growers should already adopt these practices in their routine, even if they are not close by to Xylella infected areas, because it's a good practice to be prepared when the problem comes. And so I conclude focusing on these links to website. This is the emergency Xylella, where you find regulation, communication, information on Xylella in the website of Apulia region. But we have also the website of the European Food Safety Authority for all control measures, and also we have the EPO standards and on Xylella Fastidiosa, especially for the diagnostic protocol, and also for the geographic distribution. And also we have our Ministry of Agriculture and another very important website, which is InfoXylella, which has been developed since the first finding of Xylella in Apulia by researchers, so by science, in order to contrast, let's say, the social media, which were not correctly informing the civil society of what was really happening on olive trees. So thank you, and I'm open to some questions.
Thank you so much, Anna Maria. Like, it was a very deep and actually introductory and informative presentation. Also, for me, personally, but very technical. That since I'm not an agronomist or an agriculture expert, but I'm sure that there are the information was really important for everyone here. So, but I am not seeing anything hands or the questions in the chat at the moment for now. But maybe do you have any questions, our participants have extra questions at the moment, or even if you don't have right now and you might have different questions later, you can always reach us, as Anna Maria has given her information over here, so you can contact us by any time. And I would like to mention that since OASIS project is focusing more about the implementation and processing side rather than the before harvesting, we are focused on mostly about the after harvesting side. But we all know that the harvesting, the olive oil sector and in general is not completely separate from each other. So the quality and everything is coming from the field itself. So, and especially in Turkey, we haven't seen any important incident about Xylella Fastidiosa. Spain and especially Italy has suffered from it, and they the information and also the experiences very valuable for us on that side, because since we haven't seen anything, but it doesn't mean that in the future we're not going to be seeing it. So that's why, since it's really important, we wanted to organize this webinar and this very informative content by the initiative of Siambari and Anna Maria personally wanted to increase the awareness about this very important topic considering the olive sector actually. So I would like to thank you for personally, Anna Maria, for for so much again, and also Siambari for the webinar happening. And as I said,
Thank
so
you.
I will be I will after that we will be sharing the video on YouTube channel, and I would like to ask Anna Maria if it's possible to be sharing also the presentation with our participants, if it's okay.
Yeah, I send you the PDF.
Yes, I already have
already.
it.
Yes,
Yes.
sure.
Okay,
This is
perfect.
was for the purpose to share.
Okay, perfect. So also we will be after the meeting, like when the recording is available, I will be also uploading it on our YouTube channel, and with all participants, I will be sharing our YouTube channel link and also specifically the video link. So whenever and anytime you need, so you'll be able to reach the content.